Hi, everyone. Happy Friday. So a couple announcements right here on the board. So there's no class or discussion on Monday, the holiday. You can go to a Tuesday discussion and see the syllabus for the time and rooms. On Wednesday, 6 to 7 p.m., you can make up discussion if you can't attend on Tuesday. And check BSpace if you forget all of that, and there will be an announcement there that will tell you the same thing. My office hours today, Friday, are 12:15 to 1:15. So if you want to see me today, come by PLSB 12:15 to 1:15, and I will be there. And next week, I've added a couple of extra office hours. So check BSpace. I put an announcement there yesterday telling you when office hours are next week. So if you want to come and see me next week, there'll be some extra times to do that. And then, as you know, I'm holding an exam review a week from today on the 22nd, Friday night, 7 to 9 p.m. It'll be in that big lecture hall in Dwinell. And again, we'll have this announced on BSpace. All right, so today we're going to finish up chapter 9, and we're going to talk about anaerobic respiration. And what I want to do is we're going to talk about fermentation, anaerobic respiration, and then a little bit about the regulation of cellular respiration, kind of cycling back to some of the things that we talked about earlier in my lectures about how enzymes are regulated and how pathways, like the ones we're talking about, are controlled in cells so that excess energy is not made at times when the cell has plenty of it, and conversely, where a lot of ATP can be generated at times when the cell's or tissue type needs to do that. So before we dive into that, I want to do a little recap of what some of the things we've been talking about with a couple of quick questions. So pull out your clickers, and let's do a couple of wake-up questions. Okay, and here's the first one. So take about 40 seconds to do this, and I'm going to start the timer right now. Mike, where's Mike? Okay, stop. Except that I can't find my mouse has disappeared, so I cannot do anything. There it is. Okay. Just very briefly. There we go. Okay. Okay, and then click on that. Okay, there we go. Little technical difficulty. So you can see that pretty much everybody got that one right. Okay, that was a pretty easy one. Let's try one more. Okay, and I'm going to start the timer right now. Okay, so about 6 seconds left. 3, 2, 1, stop. Oh boy, wow. I gotta show you this. It's not letting me, let's see. Okay, now it's not letting me show you the screen, unfortunately. I'd love to show you the screen because there's quite a distribution. So let's see, the highest number, about 30% of you said that the answer was D, okay, which is both ATP synthesis and oxygen consumption increase. Now let's start by thinking about what this is saying. So these are talking about drugs that actually facilitate diffusion of protons across the membrane. So that basically means that they just allow protons to go back and forth across the membrane. So remember when we were talking about chemiosmosis, what's the important thing that happens there regarding protons? We build a gradient, right? We build a gradient. So we have a lot more protons in the intermembrane space than we have in the matrix. And so if we have a drug that increases diffusion of protons across the membrane, then we tend to break down that gradient, right? So if we're breaking down the gradient, how's that going to affect ATP synthesis by ATP synthase? Decrease, I heard. Yes, and that's right. So we know that ATP synthesis is going to decrease. So as you look at the answers here, we see that there's only 2 options there's 3 options that give us that possibility, right? That maybe it's 4. Okay, so ATP synthesis and oxygen consumption decrease, ATP synthesis decreases here, and here in E. So we know it has to be either A, B, or E, I would argue, okay? And so that's that. And then the question is also asking you what happens to oxygen consumption. So how is oxygen being used in this process? Is it directly being used in ATP synthesis? Directly, it's not, right? Not directly. It's actually the final electron acceptor in that electron transport pathway. So what do you think will happen to the consumption of oxygen under these conditions? Stays the same. Okay, I heard that answer. That's right. It stays the same. So those of you that answered this question with the option E, I would argue, gave the right answer. Okay, because we would expect ATP synthesis to go down because we disrupted that proton gradient, but we expect oxygen consumption to not be affected, at least not initially. Yes. Why wouldn't oxygen consumption be affected by the proton gradient? All right, so but remember that the electron transport chain is the way that the cell is generating that gradient, right? It's not directly responsible for making ATP, right? It's actually just generating the gradient that then drives ATP synthase. Remember that really cool motor that we saw, right? So that's kind of the point of this question, is just to get you to think about the fact that these are 2 processes that are going on. They're interacting, right? They're coupled, but it's not a direct connection in a way. Yeah. Well, if you waited long enough, maybe that would happen. But initially, this is what you would expect because of the idea that these are uncoupled. They're coupled, but it's not a direct connection between the electron transport chain and the generation of ATP. And that's really the point of this question, is just to get you to think about that. Okay. So if you see a question like this on the exam, this is how you should think about answering it: is ask yourself, okay, what's happening in these pathways, and just reason out. If I block a step here, how does that affect the other things that are going on? Yeah. Right. So if you right, so your book talks about brown fat, right, and these cells that basically uncouple these pathways so that you end up generating a lot of heat rather than synthesizing a lot of ATP. Okay. Yeah, that's right. Okay. Okay, so with that, let's go on to talking about fermentation and anaerobic respiration. So these are both processes that allow cells to generate ATP without utilizing oxygen. So as you know from our previous discussion, you know that most cellular respiration requires oxygen to produce ATP. So glycolysis can generate ATP with or without oxygen, but we don't get a whole lot of it, right? Does anybody remember how many ATP molecules per glycolytic cycle pathway? How many is it? 2, right? Yeah, overall, okay? So, and that's happening with or without oxygen. So that means that under aerobic conditions, in other words, in the presence of oxygen, or under anaerobic conditions where we don't have oxygen around, this can go on, right? So glycolysis doesn't depend on the presence of oxygen. But when we don't have oxygen around, then it's not possible to couple glycolysis to the oxidative phosphorylation pathway that we talked about last time. And so when there's no oxygen around, then glycolysis couples up with either fermentation or anaerobic respiration to generate ATP. Okay, and we'll talk a little bit about what both of those are. So it turns out that some organisms actually don't use oxygen as the final electron acceptor in the electron transport chain. And these would be organisms like bacteria. So if anybody's ever been to Yellowstone National Park, for example, and you walk around and you see these bubbling pots that are yellow and sort of sulfurous, rotten egg smell. So there's a lot of very interesting organisms that are growing under those conditions, but many of those organisms are anaerobic. So they grow in the absence of oxygen, and they actually use some other molecule, it's often something like sulfate, as the final electron acceptor in the electron transport chain. So they're still carrying out respiration. They're just not using oxygen as the final electron acceptor. And so that's the sort of the fundamental property of anaerobic respiration. We're not using oxygen, but something else like sulfate as the final electron acceptor. Fermentation is a pathway that is actually using substrate-level phosphorylation instead of an electron transport chain to generate ATP. And remember that last time we talked about, we defined substrate-level phosphorylation as a process where we have an enzyme that is binding to a substrate that is phosphorylated, like this cartoon shows here, and also binding to an ADP molecule, adenosine diphosphate, and transferring that substrate, that phosphate from one substrate to ADP to generate ATP. Okay, so that's a substrate-level phosphorylation reaction. Yeah. Does fermentation generate ATP more slowly or just at lower levels, or both? I mean, the kinetics of this could, the kinetics are hard to say, right? But certainly in terms of the production of ATP, fermentation is a much less efficient way to get ATP, right? Oxidative phosphorylation is a much better way in a sense because you get a lot more ATP out. Okay, you end up fully oxidizing glucose, whereas in a fermentation pathway we don't. Okay, we'll see why that is. So sulfate, yeah, so it's a good electron acceptor. It's electronegative, but it's not as good as oxygen. Okay, it tends to be not as good as oxygen. So let's talk about the types of fermentation. So fermentation consists of glycolysis plus reactions that can regenerate NAD+, which can then be reused by glycolysis. So remember that in glycolysis, we're actually generating NADH2. And so if we didn't have a way to regenerate NAD+, eventually this would just stop, right? It would not be a sustainable kind of pathway. And there are two common types of fermentation. One is alcohol fermentation, where we're making ethanol, and the other is lactic acid fermentation, where we're ending up with lactic acid. And already, if you think about this, you can see that if these are the final products in these processes, then we're actually not fully oxidizing these molecules, right? Because we're remaining with carbon-carbon bonds, and so we're not oxidizing these all the way to CO2. We're left with these sort of intermediate compounds. So these pathways don't actually harvest all of the energy that's really present in glucose. So that's why they're actually they're less efficient in terms of generating ATP than oxidative phosphorylation. So alcohol fermentation, so that humans have known about this pathway for thousands of years. They probably didn't know the chemical details of this or the enzymes that were responsible, but certainly for millennia, humans have been using alcohol fermentation to do things like make beer and wine, to leaven bread. And these are processes that are carried out by yeast, so fungi basically, that carry out fermentation, in which pyruvate is converted to ethanol. And this occurs in 2 steps, in which the first step releases a carbon dioxide molecule, and then we're left with a 2-carbon molecule, which is, in this case, ethanol. And this slide sort of shows how this pathway works. And so we have glycolysis, where we're generating 2 molecules of ATP. That's the net yield, right? So we're generating 2 molecules of ATP. Of course, we're actually making 4 molecules overall, but we're consuming 2, right? So we end up with 2 molecules of ATP. And we also generate 2 molecules of NADH in the process. And so we end up with these 2 pyruvate molecules. And so remember from last time that these can enter the mitochondrion, where in the first reaction that gets this molecule ready to go into the Krebs cycle, or the citric acid cycle, CO2 is released, and this acetyl group gets coupled to coenzyme A, right? That's if it's going to go into oxidative phosphorylation. But what if it's not? Then what happens is that this pyruvate can actually be broken down again enzymatically to generate acetaldehyde, which is shown here with release of carbon dioxide. And then this acetaldehyde is also enzymatically converted to ethanol with the oxidation of NADH to NAD+ in the process. So it's a very nice way to regenerate NAD+ that can then cycle back into glycolysis, so this pathway can continue, and ATP can be generated this way, right? So ethanol is the end product there. Okay, and so that's alcohol fermentation. There's another type of fermentation that goes on, in which the terminal product is lactic acid. And what happens here is that pyruvate is reduced by NADH to form lactic acid or lactate as an end product, and there's no release of carbon dioxide because lactic acid is actually a 3-carbon molecule, right? So there's no release of CO2. And so lactic acid fermentation is carried out by other kinds of fungi as well as some bacteria. And again, humans have figured out how to use this productively for food production, and these organisms are used extensively to make things like yogurt and cheese that involve lactic acid fermentation. It's also interesting to note that human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. So if you're exercising really strenuously and your muscles are temporarily deprived of oxygen, they will actually use lactic acid fermentation to generate ATP. And so that's why you build up lactic acid in your muscles. So this is just showing the pathway here. So again, we have glycolysis going on. Glucose is being converted to pyruvate over here with production of 2 ATPs and 2 NADH molecules. And then again, in this pathway, enzymatically, pyruvate is converted to lactate. There's no breaking of carbon bonds here, so we have no release of CO2 in this case, but we do have oxidation of NADH back to NAD+. So this recycles NAD+, so it can go back through the glycolytic pathway. We can continue to generate ATP. And of course, this is kind of a waste product, right? So it'll build up temporarily, and then eventually it'll get shuttled to the liver where it gets broken down. So we can just do a quick comparison of fermentation to anaerobic and aerobic respiration. So in the notes for the lecture, I had just a comparison to overall cellular respiration, but I wanted to actually be a little more detailed in how we talk about this. So you can, and this is, your book talks about this, comparing fermentation to aerobic or anaerobic respiration. So these are 3 pathways that all use glycolysis to oxidize glucose and sometimes other organic fuels, like we can feed fructose into the glycolytic pathway too, right? If you look at that pathway, you can see that fructose can enter at a downstream step. And these get converted to pyruvate. And then in all 3 pathways, NAD+ is an oxidizing agent that accepts electrons from food during glycolysis and gets reduced in the process to NADH. Right? So those are properties that are shared by all 3 of these pathways. But they're different in that they have different final electron acceptors. So in the case of fermentation, the final electron acceptor is an organic molecule, right? So like pyruvate or acetaldehyde. In aerobic respiration, the final electron acceptor is oxygen, and sulfate is the final, or could be the final electron acceptor in anaerobic respiration. All right, so again, think about the fact that when we go through fermentation, we are actually not fully oxidizing the glucose molecule because we're left with these organic molecules as products. So you know right there that that's a much less efficient way of generating ATP because we're not actually taking full advantage of the energy that's stored in glucose. We leave some of those carbon-carbon bonds not oxidized, right? So it doesn't really take full advantage of the glucose molecule. Again, then respiration produces, again, your book says, we used to say 38, but if you take into account ATP molecules that are being utilized for transport of these different compounds across membranes, it turns out it's a bit less than that. So your book now says, well, it's probably more like 32 to 34 ATP molecules that are generated per glucose molecule, whereas in fermentation, we're actually only generating 2 ATP molecules per glucose molecule, right? Because we don't actually get any more ATP out of those fermentation steps that convert pyruvate to something else, but we do get to regenerate NAD+. Yeah, question in the back. Yes. Or it could. I should say, I don't know. Actually, now I'm backtracking a little bit. Mike is nodding. I think it could be less, right? Because the thing about anaerobic, right, is that oxygen is really the best final electron acceptor. It's the most electronegative of the molecules that do this. So if you have something like sulfate, it works, but it doesn't work as well. And so that's probably one reason why most respiration isn't using sulfate, right? It's using oxygen. It's just better for the pathway. Get more energy out of it. Okay, so then we can talk about different kinds of cells or organisms in terms of the kind of metabolism that they're carrying out. If we have an obligate anaerobe, then these are cells or organisms that cannot survive in the presence of oxygen. In fact, in some cases, for those organisms, oxygen is toxic to them, right? They actually can't survive. They have to carry out either fermentation or anaerobic respiration. And in the presence of oxygen, they're very unhappy or dead. And so those kinds of organisms you might find them in, again, in sort of environments, very specialized environments where they actually don't have access to oxygen. So they've had to evolve other ways of generating ATP ultimately, right? They just don't have the availability of oxygen. And when they find themselves in an oxygen-containing environment, they actually can't deal with the toxic byproducts that are made by oxidation reactions. So in contrast to this, yeast and many bacteria are what are called facultative anaerobes. That means that they can survive using either fermentation, where they don't require oxygen, or by carrying out oxidative phosphorylation through cellular respiration. Right? So they can survive either way. So they're very adaptable. And one thing that's, so think about the fact that in our bodies you can consider our muscle cells to be facultative anaerobes, right? Because they're able to function, they're able to carry out fermentation when they're temporarily deprived of oxygen, like when you're exercising really strenuously. But most of the time, if oxygen's around, they would be carrying out oxidative phosphorylation, right? So they'd be going through cellular respiration. And then if we have a facultative anaerobe, then this molecule here, pyruvate, is really a fork in the metabolic road that leads to 2 alternative pathways for breakdown. Right? Pyruvate can either be shunted into a fermentation pathway where we don't end up harvesting all of the energy that's stored in that molecule, but we're able to keep glycolysis going. Or it can, if oxygen is around, it can go, or some other electron acceptor, if it's a sulfate using bacteria, for example, then it can go into oxidative phosphorylation and we get a lot more energy being produced. And the next slide here just shows a cartoon that illustrates where this is all going on. So remember that glycolysis is happening in the cytosol if we're talking about a eukaryotic organism. And so pyruvate is made in the cytosol. And then if oxygen is around, then it will get transported into the mitochondrion and gets coupled to coenzyme A and make acetyl-CoA. And then this is the molecule that actually enters the citric acid cycle here. Right? And again, that's going on inside the mitochondrial matrix, right? That inside the double membrane of the mitochondrion. But if there's no oxygen around, then pyruvate can be fermented to ethanol or to lactate, depending on the type of organism and the enzymes that are present. And this doesn't involve going into the mitochondria. So this is all going on inside of the cytosol, generating these products and keeping the glycolytic pathway going by producing NAD+ in the process. So one of the things that's really interesting about studying metabolism like this is it turns out that we can actually learn some things about how organisms have evolved and how they're related to each other. So what's been found is that essentially all organisms